Okay, let's get started. So welcome everybody to today's Cancer Center seminar. I have the privilege and honor to introduce Professor Kathleen Barnes. Dr. Barnes has two roles on the campus. She's head of the Division for Biomedical Informatics and Personalized Medicine, as well as being director for the Colorado Center for Personalized Medicine. Dr. Barnes has been here a little over 2 years, having come here from Johns Hopkins, where she started as a junior, I think as a postdoc, and then junior faculty, senior faculty, senior leader. So now she's senior leader on this campus. She's a little bit pressed for time, so I'll keep the introduction short. She's been continuously NIH funded for a long time. She's been a leader in genetic epidemiology studies, primarily looking at asthma, has experience working with worldwide cohorts, and has been basically one of the leaders in sort of pioneering genomic data use cases for big data computing applications. So today she'll be talking about the Colorado Center for Personalized Medicine. Kathleen.
Thank you for the introduction, Debashis, and you're rushing my time here. So it's 1 year and 2 months, and I've been told I can't play the I'm new card much longer. It feels like 2 months. But anyway, thank you for the very kind introduction. And I suspect a number of you have heard me present or introduce the center. For those of you who have, to jazz it up a little bit, I want to show you some of the recent developments and directions we're moving. It's pretty exciting. I understand that for some of you, you probably haven't heard me present yet. You probably wonder what the heck personalized medicine is. Although I believe, let's see, am I able to forward, double talking, this is going to go forward, right? Okay. We're not. Or not. Here we go. Sorry about that. For some of you, probably wonder what personalized medicine is, although I understand, and unfortunately I wasn't able to be here, that a couple months ago, one of our newest faculty recruits in the center, Matt Ryoth, spoke with you about precision oncology. I'll touch on that just to give you examples. So to answer the question, what is personalized medicine? I guess it goes forward on the left. There's lots of different definitions for personalized medicine, but in essence, personalized medicine is a young but rapidly advancing shift in traditional medicine, if you will, or healthcare. The difference being that now we're taking into consideration much more information about the individual patient than we did before. So taking into consideration unique clinical, also genetics, genomic, and environmental information. And I would argue it goes much further than that. Also, through wearable devices and mobile health technology, we're also beginning to capture behavior. What Debashis didn't have time to say is that, well, he wasn't actually a Gator, but he grew up in Gator territory. When I was a doctoral student at the University of Florida, actually my PhD is in biological anthropology. And so for me, this is a very exciting time in our profession in biomedical sciences, because I really feel that in personalized medicine, we're beginning to integrate some of those basic principles of biomedical anthropology, and also considering culture and behavior much more than we ever did in the past in terms of how to care for the patient. So in essence, personalized medicine is really saying that one size doesn't fit all. And what do we mean by that? You know, some of the best examples really come from pharmacogenetics, and I'm going to talk a lot about clinical pharmacogenetics. This slide, it may be difficult to read from the back, but in essence, it's a fairly recent summary of the top 10 grossing prescribed drugs in the United States, and showing that for each one of these drugs, for each different condition, I'm sorry, cancer isn't represented on the slide, but for every individual for which the drug is effective, that is the blue individuals, there are multiple other patients for which the drug is ineffective. So clearly one size does not fit all in terms of the therapeutics we prescribe to patients for different complex traits. Again, the idea behind personalized medicine then, if you focus on the pharmacogenomics side of it, is that it's about the right dose of the right drug for the right patient at the right time. And we can illustrate that by imagining a group of patients who all have the same diagnosis And by capturing their genetic blueprint, if you will, we can then begin to categorize those patients into individuals who might be a non-responder or a toxic responder to a conventional therapeutic. And for that individual, we now have information that will guide us in treating individuals with an alternative drug that will be more effective rather than this trial and error. And of course, within that group of patients, there will be a number of individuals for whom there is no toxicity. The drug works well for the condition that they have. So again, this really falls into the category of clinical pharmacogenetics. And as I was putting the slide deck together, I was mindful of not conveying, I think, misinformation that personalized medicine is only about clinical pharmacogenomics. But as I think I can demonstrate to you, it is probably what we consider one of our low-hanging fruit and a good starting point. So a little bit about clinical pharmacogenomics. For those of you who aren't familiar, again, it's a subset of personalized medicine, sometimes called precision medicine. It primarily uses genetic information to understand inter-individual difference in drug response. It's not the only information we use, but that's the primary information, whereas you can imagine in other avenues or corners of personalized medicine, we might leverage other types of omics data. Differences in drug responses, importantly, as reflected by safety, efficacy, and side effects. Considerations in implementation of clinical pharmacogenomics are important. So we often abbreviate this PGX, and not all PGX merits translation into clinical action. And what we mean by that is that two criteria have to be met and able to move forward with clinical decision support and basically implementing decisions informed by a patient's genetic background in the clinical space. So the first would be clinical validity. That is, we have to accurately predict the presence or the absence of this relationship on what we call drug-gene pairs being related to the disease of interest, and clinical actionability or utility. That is, even if it is a valid drug-gene pair and we want to move it forward, will this information actually alter care? Can decision-making be made that will be different from prescribing the conventional therapeutic? So the question is, how do we prioritize existing evidence? This is really the nuts and bolts of clinical pharmacogenetics. We are relying on a number of resources out there. I want to underscore that this is a very fast-moving area in biomedical sciences. Resources are changing rapidly. One of the resources is the PharmGKB, or Pharmacogenomics Knowledge Base. This is a resource available that collects and curates the information about the impact of the human genome or genetic variation on drug responses. And the pyramid is illustrated here to show you this process. And a very long process reaching clinical implementation. An organization that in the field we rely very heavily on, that was established several years ago, and it was a shared project between PharmGKB as well as another organization, the Pharmacogenetics Research Network, is CPIC. And CPIC is that reference that many of us rely on at this point in time. It was recognized that we have a need for standardized guidelines, if you will, to instruct clinicians on how to prescribe therapeutics based on genetic information. It is a peer-reviewed process, and again, it's designed in cataloging this information and updating this information regularly so that institutions such as our own center can move forward with identifying these drug-gene pairs that we want to implement in the clinical space. And just an example of how quickly this is moving forward. So if you take into consideration information that's being collated through CPIC and other resources, this is a really good read for those of you who want to learn more about clinical pharmacogenomics and where we are now. It came out just over a year ago. And basically what this illustrates is the number of FDA-approved medications for which there has been a demonstrated relationship, that they're actionable according to pharmacogenes, that is, when you combine genetic information with response to the drug. So about 7% as of 2015, this number has grown, if this figure could be updated, which doesn't sound like a big number, but it is a much bigger number when you consider that for all prescriptions in the United States, for that time point when this graph was generated, about 4 billion prescriptions. Of those, about 18% fall in this category of actionable pharmacogenes, if you will. So the number is growing rapidly, and again, I come back to that metaphor of the low-hanging fruit. We probably overuse it in biomedical sciences, but we're really using it a lot these days in personalized medicine, and I would argue that one of the lowest hanging fruit are probably pharmacogenetics, specifically of cancer drugs. And many of you must have been here for Matt's presentation, and he was very kind to lend to me a couple slides just to underscore precision oncology as a great example of pharmacogenetics in personalized medicine and how we can move forward. So, for example, molecular profiling of a tumor, the first step would be to collect the biopsy. Second step, pathologic confirmation of the cancer, and then we can begin to move that forward and collect a biospecimen from the tumor, extract the DNA from the cancer cells, push this through genomic screening, genetic genomic screening, and then we could begin to make decisions, if you will, by categorizing these mutations according to the actionability. And from this reference, and again, slide prepared by Matt, we can begin to think of these categories as those that are actionable in principle. And there are several tiers of that. That would be FDA-approved drugs versus drugs that are currently in clinical trial. Certainly, another category would be prognostic. And finally, there are going to be a number of these mutations that would fall under the category of variants of unknown or uncertain significance. As further example, there are critical data domains that we rely on in precision oncology, further categorized into genotype and phenotype. And what's unique about cancer research, of course, is that when we talk about genotype, we're referring both to germline and the tumor somatic. And so considerations for genotype are critical for success, but we cannot underscore the importance enough of fine-tuning of the phenotype, which will be essential for interpretation of the genotype. We are trying to move into a space where we're not just relying on, for example, ICD-9 codes, but a much deeper understanding of the data, and all types of data are required for its success. Again, I know this is probably not an easy slide to read, but in that same review, what I really want to do by this is underscore the very large list that we have even 18 months ago of actionable somatic genetic variants in cancer cells associated with specific medications or drug gene pairs from somatic variants. And you can see quite a number of different variants that have been identified, validated, associated with response to a number of different commonly prescribed cancer medications. So certainly moving forward in that area in personalized medicine. I want to sing out for Matt, who's leading a study that's being supported by the center, going back and performing whole genome sequencing, looking for signals in melanoma. This study, we've just completed the sequencing. Matt has just completed the sequencing. But this is an example of the power of combining genomics data with well-characterized, well-phenotyped populations in discovery of better therapeutics for patients. So I want to take a step back and sort of throw the question out there, why do we really care about genetic variations to begin with? Well, I've addressed this notion that genetic variation, we know, is associated with not just our predisposition to diseases, and as Debashis introduced, my main focus has been on complex traits, but certainly we're interested in all different types of genetic traits. Environmental factors in response to drugs. So I've addressed that, but we're also, it's an academic question, why we're interested in genetic variation in populations. Of course, genetic variation dictates our phenotypic differences and so on. There's also a lot of interest, which is beyond just academic interest, in how genetic variation can tell us something about our ancestral past and human migration history. And why is this important in the field of clinical pharmacogenetics and personalized medicine? I just want to say a few words about that, and that's primarily what my personal research has been about. So it's interesting when we think about the modern human migration out of Africa over different time periods, and at the end of the day, we're really a continuum, if you will, from a genetic perspective of different race, ethnic groups, in terms of our ancestral past. And this has become a very hot topic, if you will, for companies such as 23andMe and Ancestry.com. In my experience, some folks are more interested in knowing about what village they came from in Ireland than if they carry the BRCA1 mutation. So it's of big interest, but it is beyond academic, and it is beyond just knowing about your ancestry. An area that we're particularly interested in building out in our center is to address this issue of what a patient's ancestral background has to do with their response to therapy. So as Debashis mentioned, my main area of research is asthma, and I'm just going to say a few words about this as an example of why we think this is important in the context of personalized medicine. I have been particularly interested in the health disparities of asthma, like many other complex traits in the United States, in the striking disparities of both morbidity and mortality according to race. So African Americans as a group suffer more morbidity and mortality to asthma than all other groups, with the potential exception of Puerto Ricans. And our research and others suggest that an explanation for that would be the African contribution to the Puerto Rican background. So with that as the preface, we know a lot already about common therapeutics for asthma in terms of genetic variants associated with response to the different drug classes. So leukotriene receptor modifiers, inhaled glucocorticoids, and particularly short-acting as well, and I don't show it here, long-acting beta-2 agonists. And perhaps one of the most commonly prescribed medication for asthmatics, as some of you may know, is albuterol. And in fact, if you look at the functional variants that have been associated with response to these different classes of drugs, and you then look at the frequency of those variants across different continental populations. So for those of you who aren't familiar with these acronyms, this reference population through HapMap and 1,000 genomes is a reference that we use as a tool in genetic research to tell us how common this particular variant, for example, occurs in South European, so white individuals, compared to West African, Yoruban-speaking folks from Nigeria, compared to Southwest African Americans, Mexican, Han Chinese, and Tokyo Japanese. These are common reference populations, if you will, that we use as a catalog. And what you see here is, I'm sorry I didn't highlight that for you, is for individuals of African ancestry, these two columns, YRI and ASW, some striking differences in frequencies really across the board, even being much more frequent or much less frequent than the other ethnic and racial groups. And so we know there are differences in responses, for example, to a functional variant in the ADRB2 gene associated with response to short-acting beta agonists. Another more generic example outside of asthma, if you will, would be warfarin. And this is perhaps one of the most exciting, I would argue, discoveries in clinical pharmacogenetics, arguing for consideration of ancestry in making decisions about prescribing medication. So warfarin, I'm sure, as all of you know, is a potent anticoagulant. It's commonly prescribed. Unfortunately, it's difficult to manage. There's significant interpatient variability in the dose required for efficacy. narrow therapeutic index. And interestingly, warfarin is one of the leading causes of adverse drug responses or events in hospitalizations in the U.S. In initial clinical trials, which are very exciting, variants were identified associated with dosing of warfarin, but the flaw in this original design was that the majority of the patients enrolled in those clinical trials were white or European. And it turns out that one dose doesn't fit all, just as one therapeutic doesn't fit all, that when you stratify according to race, African Americans have a different set of variants that predict the dosing of warfarin compared to white patients. So this underscores several points that we already know in biomedical science. One, non-whites are woefully underrepresented in NIH-funded clinical trials. We all know that. But that we need to begin to leverage these catalogs, if you will, these ancestry catalogs in making decisions about therapeutics for the individual patient. Admixture is something we spend a lot of time thinking about. So the profile of a continental African will look different than the profile of a European American will look different than the profile of an African American, because African Americans are an admixed population, as are others. So if you focus on the purple circles, which are African Americans from the southwest of the U.S., as well as the blue, the West African, what you see across the board, again going back to these three classes of asthma drugs, this illustrates that table I showed you a few minutes ago a bit clearer. You can see these striking differences in frequency of variants associated with those therapeutics. So admixture really matters a lot as well. Now, moving on, when we began to build the center, the Colorado Center for Personalized Medicine, we wanted to take a look at what folks are building elsewhere. We don't want to reinvent the wheel. I argue, and the reason I came here is there are a lot of really exciting opportunities on this campus that are different than other places. But we took a look at what others have done in terms of building their centers, and it's a fairly standard template, if you will, in designing a personalized medicine center. It obviously starts with the patient, and key to success is the extensive electronic health or medical record data, the EHR, EMR, collating those data. But as I'll show you, what I think is very exciting about this campus is that we've been able to collate far more than the EPIC data, for example. From that patient, in addition to the EHR, we collect something from the patient, typically a biospecimen, build a biobank. And in terms of what we collect from the patient, that varies considerably across different centers. As a starting point here at CCPM, we focused on DNA, and I'll talk a little bit about that, but bear in mind our plan, our roadmap is to greatly expand that to different types of data, RNA for transcriptome, DNA for methylome, proteomics, and so on. And so, again, this is sort of a standard template. The clinician is the direct interface with the patient, but underneath all of this is the research and discovery that's taking place to move medicine forward. If we go back to the clinical pharmacogenomics that I was discussing and think about what we can learn from other centers in terms of what they're already doing in clinical pharmacogenomics, certainly key elements there, you probably understand this now from the vignettes that I presented. First of all, we have to obtain the genomic data. We can't do anything without the genomic data. How we obtain that is critical. If we want to ultimately deliver results back to patients, that data flow has to remain clinical through its entirety. And so that necessitates developing out something like a CLIA-approved or CAP-approved biobank. Interpretation of genomic data, this is critical. We need the infrastructure in place for mining big data and the wherewithal to interpret findings. This is really, I would argue, one of the biggest challenges that we face, and that's returning results. And that's because, as you can imagine, and I'm not going to say much more about it in this presentation, there are a number of issues, ethical issues, around returning data to participants. Are they aware of what they're signing up for when they give that blood specimen to you at the outset of enrollment, that you're going to give information back? Perhaps information about whether or not you should be prescribed warfarin or not is fairly innocuous, but do you want to know if you have a variant that is associated with risk of developing Huntington's disease? These are very difficult questions, and for this within the center, we're building out an ethics component to that. At the end of the day, we want something that looks like this slide. We want to be able to generate the genomic data, combine that with the phenotype data, make a decision about whether or not a patient should be prescribed drug X or an alternative, create a pipeline for transmitting that information back into the clinical setting so when the physician is with the patient, he or she sees this pop up on the screen, in this case in Epic, helping that individual make a decision about what to prescribe to the patient. And so this is one of our primary goals in the center and what we're working towards now. Again, you can look at it as a decision tree, such as this slide. If Epic is the main mechanism by which we share information about patients, you ask the question, is there CPIC guideline for the drug? Is there genetic information? If there's neither, then you would prescribe the medication as you would normally. But if there is that information, you might make a different decision. The other thing we were interested in is just sort of general, what other precision medicine, personalized medicine centers look like. We hired a marketing firm called Global Prairie last year to do an overview of the national landscape, and we threw in the UK Biobank also as a successful example of a personalized medicine program associated with an institution. We went through a filtering process outlined in this slide. We started with a full list of centers that are part of the Personalized Medicine Coalition. At the time, there were about 70 registered. We went through a further filtering process of which ones have been most successful in leveraging their program to generate NIH funding. This is certainly a goal of our own center. If we build it, you'll come. If we build it, you'll be able to leverage the data that we generate to ask new research questions and apply for NIH funding. We also added partners because I had worked closely with that group in the past and was impressed by their center, and we wanted to see how all of this fell out. So here's the list of personalized medicine programs that we looked at closely. In our industry overview, if you will, what we were particularly interested in, as others are building their personalized medicine programs, I've given you some great examples of clinical application of personalized medicine, but again, we also want to build a program that's going to allow us to grow in terms of research and discovery. And so at the end of the day, to what extent are these different centers that we looked at integrated in terms of clinical application versus research focus? So this is where we want to be. We want to be a fully integrated center that takes equal consideration into clinical application and research and discovery. And this was how the different sites fell out. And it was very interesting to us. So when we're thinking about, again, not reinventing the wheel, how to build out our own pharmacogenetics program, we can learn a lot from those programs who have really focused extensively in that area. But again, we want to keep in mind the research opportunities as well. So a little bit about us and what we look like and what we're building. So the Colorado Center of Personalized Medicine, we have a website that we're about to launch, and we'll make that available to everyone. We've put a lot of effort into the marketing of the program, both in terms of staff, faculty, and providers here on campus, but also the community at large. And we have a lot more to do, but one of the visions is to create a video that will run constantly around UC Health and Children's advertising our program so that people understand, A, what is personalized medicine, and B, that we want them to be part of the program. I left a slide out for the sake of time, but last year I was able to take a step back and say, what is our potential? What is our potential here at the Anschutz campus in terms of how big a biobank we could build? And if you look at the population density and you look at the number of individuals in the state of Colorado and surrounding states that are served by UC Health and Children's, it's somewhere around 500,000 new patients per year. That's a staggering number, and that's our goal. So to build the program, first we need the infrastructure, and I'm going to walk you through very briefly the different operational units that we've put into place to make up that infrastructure. That is, creating bringing together the data, generating data, and creating the tools for the research and discovery. Discovery and development is key. So once we have this information in place, we need the faculty base and partnering with researchers such as yourself for that discovery leading to new therapeutics ultimately, and then implementation of personalized medicine into the clinical setting. So there was if you're interested or you didn't see it, there was a nice story that was done on our center in the Vision last year in the 5280 magazine. And I want to just underscore a couple points here. When people say, Kathleen, why did you leave 23 years at Johns Hopkins to come to the University of Colorado and build this center? I've already mentioned the patient base, which is huge, and I would argue very different from some of the other centers, particularly the urban centers, where there's a lot of competition from other institutions. One of the biggest carrots is the institutional support here. So this figure, I believe, is already outdated considerably, but as of about 6 months ago, when I first made this slide, the institution had committed over $80 million in building this program. What's exciting about that is that the stakeholders cut across the institution. So it includes UC Health, Children's, UPI, which is now Colorado Medicine, obviously the university itself, School of Medicine, Department of Medicine, big stakeholders, and beyond. So there's incredible commitment. I've talked about the mission in so many words. It's obviously to advance personalized medicine back in the clinical setting and to leverage the data that exists here. Compass, and I'll talk a little more about that, was an initiative started actually several years ago, I would argue somewhat before its time compared to other programs, in pulling together all of the EPIC data and other types of data and putting that in one place to work with. And the goal, obviously, at the end of the day is to improve healthcare in the clinical setting. What have we built so far? So this may be, again, a bit difficult to read, but this is the organizational structure of the center as we have conceptualized it so far. By the way, I want to send a call out to Dan Theodorescu, who I met with at the end of the calendar year. And one of the things that was really important to me when I first arrived here was thinking about how we can build the bridges stronger between the Cancer Center and CCPM. It's an obvious fit, which hopefully I've been able to demonstrate for you just in the area of clinical pharmacogenomics alone. Our structure is such, so we're based over in Building 500. Just in the last 6 months, we've added considerably to the staff infrastructure. We have a really strong finance and administrative staff in place. The division, as Debash has mentioned, so I'm the head of the Division of Biomedical Informatics and Personalized Medicine, which is one of the divisions in the Department of Medicine. This is intended to be the academic home for the faculty that we recruit into the center, of which we've recruited a number so far. And then one of the goals, and a call out to all of you, is the original mission was to also build that faculty base through secondary appointments here on campus and other campuses. We've been working closely with the BioFrontiers folks up at Boulder to build bridges there. But the goal is to have something on the order of 20 to 30 secondary appointments. And so I see this as a real opportunity, partnering with the Cancer Center, also in joint recruit of primary faculty. I'm going to talk a little bit about Health Data Compass, the computing center that we needed to build to make this succeed. A few words about the biobank. Clinical operations is, again, I would argue, one of our biggest challenges. I had no idea until I was 6 months here that there's something on the order of 285 outpatient clinics alone at UC Health. So getting your head around how do you capture potential participants at point of entry is complicated and something that we spend a lot of time with the clinical operations folks over at UC Health working out. I'll say a few words about molecular diagnostics in terms of partnering with pathology and Komoko and expanding that out into a heritable disease component. So this is what the operational units look like by illustration, and we'll start with the data warehouse, which many of you, I think, already, most of you, probably know a lot about Compass, because of Compass has worked very closely with the Cancer Center and predates my coming to the institution. This is a really unique concept when you look nationally at other personalized medicine centers. It's not to say that other centers haven't done something like Compass, but I think it was very foresightful for leadership to put Compass in place even before the center was initiated. So it was first recommended in 2011. It was launched in 2014. And this is an overview, if you will, of this notion of blending different types of data for different purposes. So if we just focus on the personalized medicine box over here, it really is this integration of Epic data at the different healthcare provider organizations, care analytics, research operations, research analytics, all of these data coming together in the space that is called Compass. And beyond personalized medicine, there are other purposes for collating all of these different types of data, and all of this is actually currently underway. This is a fun little test of the utility of Compass. So Michael Kahn, a few months ago, a researcher came to Michael and said, we want to ask the question, is there a correlation between legalization of marijuana and the rate of facial fractures at UC Health? And within very short order, because all of the data are already there, they were able to answer the question. The answer is yes, there is a correlation. This is kind of a silly example. Of course, there are much more in-depth research projects that can be and are underway leveraging Compass. I mentioned at the outset, of course, a critical component in a successful personalized medicine program are the biospecimens, to be able to generate the genomics data and other types of omics data. And for that, we need a state-of-the-art biobank. Again, we're focusing initially on DNA because DNA is easy and it can tell us a lot. This was built out last spring. We moved into the space in Biosciences 2 across the street on the 3rd floor to build a biobank that could handle a capacity of up to 500,000 samples per year. Again, the focus initially was on extraction of DNA and high throughput genotyping, which I'll say a little bit about. This will be expanded, and plans are already underway to expand this, and plans are already underway to expand the type of material that we collect from the patients. So biobanks are interesting beasts. Many of the biobanks in those nationally recognized established personalized medicine programs collect a sample and store it, waiting for the researcher to come along and ask a question of that sample or other samples like it, and then something happens to the sample. It's an expensive way to manage a biobank, and we argue that a much better approach is to generate data on that sample the minute it hits the laboratory. And so we were able to partner with UC Health and negotiate with Illumina to come up with a process, something that we could do to that sample the minute it hits the door. Now, the question I get asked all the time, because everyone hears about the $1,000 genome, is, aren't you going to do whole genome sequencing on every sample that hits the door? And, you know, in the field of genetic epidemiology and statistical genetics, this is a hot topic. At the end of the day, we ask ourselves, what is the utility of all of those data on so many samples? With a really well-designed SNP chip, we can have almost as much information, admittedly not all, but almost as much information for a fraction of the cost. So the current price for whole genome sequencing never really comes to around $1,000. It can with a lot of negotiation, but consider even $1,000 versus $70 a pop. So for that reason, we went with something called the Multi-Ethnic Genome-Wide Association Array, or MEGA array. I think some of you in the room have been very much involved in this process and contributing to some of the content. I was excited about this array because back when I was at Hopkins, we had an NIH funding to come up with a SNP chip that was specific for populations of African ancestry because, as some of you may know, many of the commercially available SNP chips, all of them until really recently, were very biased in European ancestry populations. And so we failed to make the discoveries we needed to make on these commercial chips because they just weren't designed with African populations in mind. We designed an African diaspora power chip in partnership with Illumina after whole genome sequencing 1,000 individuals. And subsequent to that development, we partnered with the PAGE Consortium, an NHGRI consortium, that was also making a customized chip focusing on the 26 populations from the latest phase of 1,000 genomes, was much more gene-centric, exome-centric. We came together and developed the mega chip, and that's just under 2 million SNPs. And the beauty of the mega chip, besides, and ironically, of all these populations, continental and subcontinental populations, the chip actually performs best for populations of African ancestry. Besides being a very dense GWAS chip, if you will, it's been enriched for content that includes a number of exomic variants, known functional variants, and so on. It also allows for considerable customization. So we can add up to 300,000 of our own SNPs that weren't already represented on that chip. And to do this, after signing this agreement, we issued an RFI on this campus, reaching out to investigators on campus to say, what's your favorite disease? What's your favorite gene? What are the SNPs in that gene, or the range, or the locus in that gene that you want covered on the Colorado signature mega chip? And so that was added to the chip. So we have 32,000 of these chips sitting in the biobank, ready to be run as soon as the biobank, as we complete the CLIA registration of the biobank, which we estimate to be somewhere around the beginning of March. So we pull all of these data together, the phenotype data, in this case the genomic data. How do we bring all of that together? A challenge that we faced when we started the center was that there was no centralized place, if you will, computing center that could handle this much data, because you're talking about a lot of data. Zhu Fang in our group, who directs the operational unit I want to talk a little bit about, created this image, which I think very nicely summarizes the different types of data that we're working with and the flow of data out of the biobank, and then there's Encore, which I haven't even discussed. And so essentially we are here with something we call TICKR. So TICKR is short for Translational Informatics and Computational Resource. I should mention that Debashis is the official director of this part of the center, has really led the charge in developing this out. In essence, we had three goals, if you will. One, we needed the infrastructure for which we could analyze all of these data. A high-performance computing exchange, we modeled that after several institutions. We fondly refer to this as ROSI, after Rosalind Franklin. And so this is a resource, and I'll say a little more about this, but essentially on February 14th, this is a resource that will be launched to the institution at large. Everyone will be able to take advantage of this as a resource. It's robust for the computing, networking, and storage that we need to handle big data. The other thing that was important to us was to develop the tools that we need, pushing that through to molecular diagnostics to fulfill our goals in the clinical setting. And so TICKR is a place where this is also taking place. And we wanted to create that, the people power, if you will, the intellect that one needs to conceptualize a study, design a study, analyze the data, and so on. And so we've built it out to fulfill all three of those needs. When you think about all of these different pieces of the puzzle, if you will. The flow looks a little bit like this in terms of the center and where everything fits and the type of data that's coming in and flowing. But again, I want to underscore, I'm really being very narrow-minded in this presentation to you in focusing on DNA and genomics information. Ultimately, we want to expand that. This is a little dated. Initially, we were testing all of this, if you will, in select clinics over at UC Health. Our recruitment has gone, it went institution-wide on October the 12th across UC Health. We have plans later in the calendar year to move to UC Health North and South and some of their newer affiliates. We're already beginning to partner with the VA and other programs on campus, such as the School of Dentistry. We're working closely with Children's to get Children's Hospital on board, and we're also in conversations and partnering with Denver Health. So we're really casting the net wide. So a little more about the biocomputing and discovery, and so how do we take all this information and really deliver this into the clinical setting? I want to just touch on 3 areas that are some of our primary areas of focus right now in the center, but again, we're growing and this is broadening quickly. So I've talked to you a lot at the outset of the presentation about clinical pharmacogenomics. Again, we see this as low-hanging fruit, and so this has been one of the first working groups that we put together. I wish I could take credit for coming up with this really clever acronym for the committee, PICCOLO, Pharmacogenetics Implementation Committee of Colorado. It's being led by Chris Aquilante in the School of Pharmacy and Dave Kao in the Department of Medicine, School of Medicine. Initially, our vision was for this committee to serve as an advisory committee to myself and the steering committee, but it's really taken on much greater life since we formed it last summer. And again, it's the interface for all of these different enterprises here on campus. The main charge of the committee is to implement, at the end of the day, implement in the clinical setting valid and actionable PharmGX drug gene pairs, starting with UC Health, but obviously, ultimately, we want to expand that. This is the roster, which is already outdated and growing. Within the committee, then, we've so far put together 4 working groups, so it really dovetails nicely into what's happening in the biobank and making sure that we're following regulations and so on so that we can give information back, clinical information back to patients. Just identifying those drug gene pairs is a challenge. So in essence, we're looking at the content already on the megachip, selecting a subset of that content for known and valid drug gene pairs, validating in our biobank, generating the data, the genomics data through the megachip, and then ultimately that information has to get, as I said, back into EPIC. So we have a whole working group just focusing on that pipeline of getting information back into the clinical space. For clinical decision support, we have a working group for that, how that's distributed, how that language actually works in the clinical setting. What I haven't mentioned here, in terms of the drug gene prioritization, again, we're focusing initially on pharmacogenomic targets, but we also ultimately want to include heritable traits that are also captured on the megachip. So this group right here is really divided into 2 different working groups. I mentioned at the outset the interest in genomics data for non-clinical purposes, that is ancestry. And hopefully I've been able to demonstrate to you why ancestry is more than just knowing about your family history, but it actually comes full circle in terms of the decisions, the clinical decisions we make. So we have just brought a new faculty member on board, Chris Janou, comes to us from Stanford, who was actually with 23andMe for some time. And Chris is going to build a center within the center called PAIR, Population Ancestry Information Resource, where we leverage the information generated from the megachip initially. Ultimately, we'll be able to leverage other types of genomic resources to really develop, if you will, a report that we can give back to each participant that's non-clinical, that says something about their ancestry we think there will be a lot of interest in this. At the same time, obviously, we'll be creating a resource from our patient population here in Colorado, which is unique to other patient populations. When we think about the demographic breakdown across Colorado, unique aspects of underrepresented, underserved populations, such as Hispanics, American Indians, really delving into that urban-rural dichotomy. I don't know how many of you have heard, but it was just reported a couple days ago that possibly one of the biggest health disparities in the United States right now is urban-rural, not as much underrepresented populations as underserved. So we think that this will be a really important development for our program, and I want to throw that out there, that this can serve as a real resource to each of you, depending on what kind of research that you're doing. A few words about molecular diagnostics, because at the end of the day, all this information and discovery needs to be translated into some tool beyond the megachip that can be applied back into the clinical setting. We've made great headway in this area. We started with Komoko, which already existed before I got here, which is based in the Department of Pathology, and partnering with Dara Eisner and Ann Thor and others in Komoko, we've been able to continue to build out the somatic component of Komoko, if you will. We then brought on board a co-director of molecular diagnostics in the center, Christy Crooks, came to us from UNC, and Christy is building out the germline side of Komoko, if you will, for heritable disease testing. I know many of you are already familiar with this enterprise and have worked very closely with Komoko long before the Personalized Medicine Center took off. I just want to, call out, there's been a lot of development, particularly in assays that have been developed by Christy and her team just in the last 6 months, where the testing can now take place here at Anschutz instead of farming that out to other vendors. A word about where we are. We're just a floor down from you at the Cancer Center based in Building 500. I'm very excited to say that there are plans in the works for a building dedicated to the Personalized Medicine Center, I'm told within 2 to 3 years. It's not going to look like this anymore. It's going to look like another RC2, and that will be behind, I guess, to the west of the current RC2. We're very excited about that. So ultimately, we will be moving into that space. Come and see us sometime. We're very excited about our new space on the 5th floor that was kindly provided by Dean Riley, and we moved in there at the end of August. Lots of space for, and this was all deliberate, we wanted to bring together the bioinformatics. So right now, Compass is housed next to Ticker. In the new building, we'll be partnered with Biostats and Dabash's operation. Lots of space for thinking and designing studies, so I invite you to come by. Who we are, I've mentioned a little bit about faculty recruits. I've been charged with building a primary faculty base of 10 new primary faculty, and as I mentioned, 20 to 30 secondary. We've been very fortunate to bring in tremendous talent, not just what was already here on Anschutz already. We had, I think, 3 or 4 faculty who moved from other divisions or departments as primary in the center, but recruiting, Matt's a great example from Vanderbilt, a FEWAS expert from Vanderbilt, a couple full professors in statistical genetics from UNC. We have Chris joining us this week for the first time from Stanford, and a couple other recruits underway. So we're very excited about this. But I do want to emphasize in building more interaction between the Cancer Center and the Personalized Medicine Center, there's lots of opportunity through secondary appointments, and I'd love to discuss with any of you who are interested. So I'm sorry I didn't leave a lot of time for questions, but I wanted to give you an overview of the center, who we are, what we're building, what our focus is on initially, and hopefully I've inspired you to think about how we can do more together between Cancer Center and Personalized Medicine. If not, come and visit us on the 5th floor. Thank you.